Hey y'all, today's the day I go over antigen presenting cells and MHC classes, right? Classes 1 and 2, right? Okay, well, so MHC class 1 is pretty simple, right? MHC class 1 is actually on all nucleated cells, right? So whether that's skeletal muscle cells, right? Cardiac cells, neurons, right? Anything with a nucleus. So anything but red blood cells, okay? Right? And here, in these asterisks, I have important facts, okay? That I'm just gonna note for you guys. So, right here, MHC class 1 is stored in the endoplasmic reticulum, or the ER, right? And that'd be right here, usually connected to the nucleus, right? Interferons, alpha, beta, and gamma, usually lead to increased MHC1 expression, right? And you'll see why this is relevant. The next one. MHC1 usually presents viruses, right? So this all ties together because whenever you usually have a viral infection, your body responds by releasing interferons alpha and beta, which would lead to increased MHC1 expression, which would lead to increased uptake of viruses and viral killing, right? Cool, isn't it? All right, anyway. So now I'm going to go over the steps, and while I'm going over the steps, I'm actually also going to, draw it in this little diagram as well, okay? So step 1, the cell is infected, right? The cell gets infected and the virus or the viral protein is going to go into the nucleus and it's going to integrate with our DNA, right? After it integrates with our DNA, it's going to produce viral RNA and DNA products, right? And these are going to be free floating in the cytoplasm, right? But these viral RNA and DNA products, they're actually called defective ribosomal products. And this is because they're not self products, right? It's made by the virus using our machinery. Now, what's the fate of these defective ribosomal products, right? They're targeted to be destroyed or broken down. And who does that targeting? Ubiquitin, right? You heard of that before, right? Yes. Ubiquitin. I know this is green. I'm going to just do this just because, it's good in the sense of viral infection. So ubiquitin 1 will bind these viral proteins, right? One. And then after that one binds, it'll start polymerizing. More will bind, more will bind, more will bind, until it comes in contact with proteasome. Now, the proteasome is kind of like a stack of tubes. And it's going to take the ubiquitin marked antigen protein foreign product. It's going to take it up and it's going to break it down, right? And it's going to break it down into one of these vesicles with a bunch of antigens instead of just like antigenic chain, right? Antigenic peptide, right? So after the proteasome breaks it down, guess where it goes? This vesicle goes straight to the ER, right? To find its MHC1, right? And the way MHC1 and the viral antigens connect, right? It's whoever has the greatest affinity, whoever creates a complete seal, a lock and a key type of mechanism, right? So these viral antigens are going to travel to the ER trying to find its MHC1 complex that it can bind to, right? And when it finds it, it's going to be transported Oh, I'm sorry, y'all. This MHC1 complex with its antigen is actually going to travel to the Golgi via another vesicle, right? After Golgi does some preparation, yada yada, and it sends that straight to the cell surface. Now, step 5, what's important about this cell surface? I'm going to tell you real quick. It interacts with CD8 T cells. The reason this is significant is specific to the way that viruses infect the cell, right? They infect the nucleus, they corrupt the nucleus by implanting their own DNA into our genome, right? Once they corrupt that, and by the way, that corruption of your genome, that's one of the mechanisms as to why some viruses can cause cancer, right? So, or more likely to cause cancer, sorry. And it releases these defective ribosomal products free, and it's free floating all throughout the cytoplasm, right? The way that viruses kill any nucleated cell is called a cytopathic effect. It's where the virus builds up and produces so much of their viral antigens to wait till it's so much to release that they end up killing and lysing the cell so that they can release that, right? So they build up as much as they can in order to kill the cell and release their viral peptides, right? So how do we avoid that? These CD8 T cells, they'll bind, and I'm going to go over this binding later in the video after MHC class 2, so just give me a second. After it binds, the CD8 T cell will actually release perforin and granzyme B, right, and cause apoptosis of these cells in order to kill these viral antigens that are already in the cell and prevent the cell from replicating, without the infection anyway, because the DNA is already corrupted. So there's no choice but to kill this cell, cause it to undergo apoptosis so that, you can have healthy cells moving forward. And then an adjacent cell will end up filling in the space and you'll be all good, right? Cool. So now let's go over MHC class 2. All right. Or sorry, before we move there, I just want to say this right here. It usually presents viruses, but not always, right? It can be intracellular bacterias like chlamydia, right? Or intracellular protozoa. Now, I would say an intracellular protozoa being like what causes malaria, which is Plasmodium falciparum, but that infects red blood cells, and we already went over that MHC class 1 only is present on all nucleated cells. Red blood cells don't have nuclei. All right, start tying in everything. Let's move on to MHC class 2 now. All right, so a couple factoids. MHC class 2 is stored in the late endosome of professional antigen presenting cells. Now, what are professional antigen presenting cells? These professional antigen presenting cells, right? These are going to be your macrophages. These are going to be your dendritic cells. Macrophages, dendritic cells. And these are going to be your B cells. Right? These are known as professional antigen presenting cells because they have, you guessed it, MHC class 2 and MHC class 1 presented on their cell surface, right? That's the reason they're called professional antigen presenting cells. So if you see that word and you're confused as to what they mean, this is what they are, okay? And HLA DM and DO haplotype deficiency or mutation would lead to dysfunction of MHC class 2. Now, the reason that is, let me go over that now, is because these proteins actually have a function in removing the CLIP protein. The CLIP protein is what is initially bound to MHC class 2 inside of the late endosome before it's even presented viral antigens, right? It keeps a hold of that binding site, and in the presence of the correct antigen, these molecules will remove CLIP, right? So MHC2 plus CLIP released from the ER, travel to the late endosome, CLIP removed in presence of correct antigen and HLA, right? HLA, DM, and DO. Okay. So MHC2 usually presents bacterial antigens. This is usually, it's not always, but let's keep it going, all right? So step one, we got endocytosis of the organism, okay? So you endocytose that bacteria, right? Via LPS, which is a toll-like receptor 2, I believe, or a TLR4, which I think is gram-positive bacteria. It's going to end up endocytosing this, and this early endosome or this phagosome already has some acidic, a little slight acidity, some slight proteolytic activity. So it's actually going to start breaking down this bacteria as soon as it comes in. So you're going to have some broken down peptides, right? Step two, it's going to break it down, right? Now, these peptides, they have to come in contact with MHC2, right? Remember, which has the CLIP protein on it. So what's going to happen? Well, it's actually going to end up fusing, right? It's going to end up fusing with the early endosome or with the endosome that has the MHC2 and the CLIP in it. It's going to fuse, and that forms the late endosome, right? This late endosome has MHC2, CLIP protein in its binding site, as well as bacterial antigens. But the late endosome also has HLA, DM, and DO. Those little two black lines. Sorry, I didn't make it any better to distinguish. But this DM and DO is going to help remove CLIP in the presence of this correct antigen. It's going to help remove CLIP. And after that removal, it releases the MHC2 product and its correct antigen onto the cell surface. Now, we discussed MHC class 1 gets in contact with CD8 T cells and activates them, causing cytotoxicity. Now, this is going to the surface to activate CD8 CD4 T cells. These are called helper T cells. Now, helper T cells have a bunch of differentiations to go between. You got Th1, Th2, you got Th17, and also follicular helpers. But I'll go over that in another video, okay? Just shortcut and sweet. It's these CD4 T cells, after coming in contact with this MHC2 and this antigen from these professional antigen-presenting cells, it's going to release cytokines, right? And these cytokines could, activate the macrophage to take it further, right? Differentiate into M1 or M2 or, all that stuff. It could cause B cells in order to activate releasing antibodies, IgM, IgG. So its primary function is it's going to release cytokines. These cytokines can cause fever, vasodilation, runny nose, congestion. Like, it's what makes the body go round, right? It can activate other inflammatory cells and all that. So that's it for the MHC class 1 and class 2. I did say I was going to go over the receptors, so let me do that right now. I have to do this. Dang it. Okay. Hold up. Oh, got it. All right. So before, okay. So I was gonna say the board changes, but anyway, um, before that, before I move on into receptor communications, I want to say that we went over interferons alpha and beta, leading to, released during early viral infection, which can lead to increased MHC expression, right? So, interferon gamma release can also lead to macrophage activation, right? Which could lead to cytokine secretion. It could lead to granule release. It could lead to activation of other macrophages, differentiation into M1, M2. We'll go over that in the macrophage video. But it can do a lot. It can lead to a lot of downstream effects in the body. All right. So let's go over receptor communications, right? I kind of tried to break it down as much as possible. It may look a little confusing. Hopefully it's not after this video, right? So I gave you guys a little key showing you which receptors are which. Let's go over the communication between MHC1 and CD8 T cells. All right. So MHC class 1 would bind to the T cell receptor, right? They bind to the T cell receptor. Okay. So the receptor communication that's occurring here is MHC1 is going to bind to the T cell receptor, right? It's going to bind to the T cell receptor on the CD8, right? And then there's something called a B7 also on the MHC1. So on all nucleated cells, they have a B7, otherwise known as a CD80. B7-1 is CD80, B7-2 is CD86, right? And this B7 or CD80 is going to bind to CD28. This is what we call a co-stimulatory binding. I'm going to say co-stim. Co-stim. It's going to co-stimulate this reaction in order to make sure that this antigen properly activates the CD8 T cell to destroy the cell, right? So it's going to co-stimulate the binding of this in order to cause its cytotoxic effect, right? All right, let's move on to MHC2. So MHC2 is actually going to bind the T cell receptor as well. Another molecule on the professional antigen presenting cells is going to be CD40. It's a CD40 receptor, right? And it's going to bind its ligand, CD40 ligand, right? And the CD40 ligand is going to be on the CD4 T cells. So CD40 ligand, CD4 T cells, right? Maybe that'll help, I don't know. But this CD40 and this CD40 ligand, can you guess what that is? It's a co-stimulatory action, right? So it's going to stimulate this CD4 T cell to release a bunch of cytokines to activate other cells and attract other cells and yada, yada, yada. What cytokines be doing, right? So that's the normal binding of both MHC class 1 with CD8 T cells and MHC class 2 with CD4 T cells. Now, let me go back to MHC class 1 and talk to you about an inhibitory binding, right? It's going to be very quick. Just bear with me. So this little guy right here, this is called a CTLA4. Now, CTLA4 is actually an inhibitory protein which binds, which will bind B7-1 or B7-2, right? It'll bind it instead of CD8, right? And when you bind the CTLA4 instead of CD8, this actually causes an inhibitory effect instead of co-stimulatory. And this might be the case of maybe like it's a self-antigen, or if you have continued inflammatory response and you don't want to shut that off, right? So do what you will with that information. I'm done with this video. Hope y'all enjoyed the content. Hope you learned something. And I'll see you next time. Peace.